A behavior environment information loop drives sensory navigation.
Have you ever wondered how a tiny bacterium can navigate toward food just by sensing chemical gradients? Movement actually alters the signals it receives next, so navigation is a two-way conversation between sensory input and action.
That sounds like a feedback loop rather than a simple feed-forward response.
Exactly. Traditional studies have focused on how signals drive behavior, but less attention has gone to the reverse path, how the organism's own movements influence future sensory cues. The authors argue that this reciprocal flow is essential for efficient navigation and propose a framework to quantify it directly from experimental trajectories.
So they're measuring both directions of information exchange? That must be tricky.
They use an information-theoretic approach, building on mutual information but splitting it into two-directional transfer entropies, one from signal to action, reactive, and one from action back to signal, active. By doing so, they can see how much the organism learns about its environment through its own motion.
And what's the payoff? Better understanding of navigation across species.
Precisely. They test their metric on data spanning bacteria, worms, and flies, over 5,000 hours of bacterial tracks, hundreds of hours for worms, and more than a dozen hours for flies. Their predictions match observed performance across these diverse systems.
That's impressive. It suggests the same underlying principles might govern navigation from microbes to insects.
And that is the core message. Bidirectional information flow underpins navigation efficiency regardless of scale or organism. To get intuition, they start with a minimal bipartite Markov model inspired by bacterial chemotaxis. Think of two behavioral actions, run, R, and tumble, T, and two environmental states, up, U, and down, D, the gradient. Each combination forms a node in a graph.
So 4 nodes total. How do they move between them?
Transitions are governed by rates that capture key biological facts. During tumbles, the organism reorients quickly between up and down at rate lambda. When running, it changes direction more slowly, scaled by a factor alpha between 0 and 1, reflecting persistence of motion.
So alpha is less than 1 means runs are longer in one direction than tumbles.
Right. Also, the run duration depends on whether the organism is moving up or down the gradient. Gamma U for up runs, gamma D for down runs, with gamma U typically smaller than gamma D, so that runs are longer when heading toward higher concentration. Tumble to run transitions happen at a baseline rate kappa, which they set to 1 for convenience.
How do they quantify performance in this model?
They solve for the steady-state distribution over the 4 nodes and compute 2 metrics. The chemotaxis index delta, defined as the difference between probabilities of running up versus down, pi RU, pi RD, which reflects average speed toward the gradient, and tumble bias terabytes, the total probability of being in a tumble state.
So higher delta means better navigation.
They then explore how delta depends on sensory gain K, essentially the ratio gamma Ds or gamma U, and on alpha. The analytic expression shows that increasing K always improves delta, while decreasing alpha, that is making runs more persistent, also boosts performance.
That makes sense biologically. More sensitive to gradients and less jittery running should help.
They confirmed this with stochastic simulations across a wide range of parameters, matching the analytic predictions. Importantly, the model's asymmetry in transition rates between sensory states conditioned on action versus actions conditioned on sensory states creates an information loop that drives navigation. The authors then connect the minimal model to real data by formalizing how much information flows in each direction. They consider time series of sensory inputs, ST, and actions at, and use transfer entropy, a directed measure, to quantify how knowledge of the current sensory state reduces uncertainty about the next action, reactive flow, and vice versa, active flow.
Transfer entropy sounds heavy. Do they compute it exactly?
Not directly. Full history transfer entropy would require estimating probabilities conditioned on entire pasts, which is infeasible with limited data. Instead, they use one-step transfer entropies that look only at the immediate next step. These are upper bounds, but still capture the essential directional information.
So what do they find in the minimal model?
They derive explicit expressions for the one-step rates using the transition parameters. The reactive flow depends on the difference between gamma D and gamma U, while the active flow scales with 1 alpha. Asymmetry in run-to-tumble transitions fuels signal-to-action information, whereas environmental persistence boosts action to signal information.
That's neat. So both asymmetries matter.
They then perform a perturbative expansion around the limits of weak control and high noise to show that chemotaxis performance delta scales with the product of these two flows, specifically as the square root of their geometric mean. This leads to an information-theoretic predictor phi that combines both reactive and active contributions.
Does phi predict real data well?
Yes. Across simulated data from the minimal model and various computational models, phi correlates strongly with delta, r squared approximately 0.95. When applied to experimental trajectories from bacteria, worms, and flies, phi again outperforms alternative metrics like mutual information or entropy production.
So the key is that navigation requires a loop. Signals inform actions, and actions generate informative future signals.
Without reactive flow, signal to action, navigation collapses. Without active flow, the organism can still bias its motion but cannot exploit environmental correlations. The balance between the two drives efficient navigation across scales.
So, Jamie, the paper just dove into a bunch of different creatures: bacteria, worms, flies, and even an RL agent. How do they all fit together?
They take each system and ask the same question: does the way information moves between what the organism senses and how it acts predict how well it finds its target? For bacteria, that target is a chemical gradient. For worms, a Gaussian scent field. For flies, an odor plume that flickers like in real turbulence. And for the RL agent, it's a reward signal.
Okay, but they talk about binarizing sensory inputs. Why make everything just on or off?
Binarization simplifies the data. For bacteria, worms, and the RL agent, they reduce the sensed chemical concentration to two states, up gradient or down gradient. For flies, the intermittent odor signal is turned into either present or absent based on a threshold. This makes it easier to count how often you jump from one state to another.
And then they talk about Markov states for behavior. That sounds fancy.
It's a way to turn continuous motion data, like speed and turning angle, into discrete categories that still carry the most predictive power about future movement. Imagine watching a bird. Instead of recording every feather flap, you just note whether it's gliding or flapping. That's the idea here.
So they end up with binary streams for both sensory input and action. What happens next?
They calculate transition probabilities between consecutive states in these streams. From those probabilities, they compute two key numbers: the reactive information flow, how much the organism's actions tell you about what it senses next, and the active information flow, how much its sensing tells you about future actions.
Sounds like a two-way street of data. Do they use each trajectory separately?
Not really. Individual runs are short and have few state changes. So they pool many trajectories together to get reliable estimates. That's why the paper mentions finite size corrections and ensemble averaging.
And how does that relate to performance? Like, is a high reactive flow always good?
They plot performance, like drift velocity for bacteria or fraction of successful odor source hits for flies, as contours over the plane of reactive versus active flows. For each dataset, they pick representative trajectories from different regions of that plane and show how their actual paths look.
So you can see if a bacterium is more reactive or active, and what that means for its navigation speed?
In the bacterial case, they found some bacteria that were more reactive, meaning their behavior was tightly coupled to sensory changes, and others that were more active, meaning their actions had a strong influence on future sensing. Both strategies can yield similar drift velocities, but the underlying information dynamics differ.
That's pretty cool. Do the worms and flies show similar patterns?
Though the specific behaviors differ, worms exhibit steer and pirouette, while fruit flies use a walk and stop strategy. The framework still captures how information flows in those systems and links it to their navigation success.
And the RL agent? How does that fit into the picture?
For the RL agent, the reward signal plays the role of the sensory input. They measure accumulated reward as performance and again look at reactive versus active flows between reward and action streams. It shows that even in a purely algorithmic setting, these information measures can predict how well the agent performs.
So the big takeaway is that this bidirectional information flow framework works across very different biological and artificial systems?
That's right. By reducing everything to binary sensory and action streams, and then looking at how information moves back and forth, they demonstrate a unifying way to predict navigation performance. It's fascinating how a simple idea, tracking how much one stream tells you about the next step in another, can explain so many different organism strategies. The authors even show that their predictor phi captures performance across bacteria, worms, flies, and an artificial agent with remarkable accuracy.
And it all hinges on those two directional flows: reactive flow from signal to action, and active flow from action back to signal. When both are strong, the organism can adapt its behavior in real time while also shaping future sensory input.
If engineers can embed a similar bidirectional information loop into their algorithms, they might achieve more efficient and robust navigation without having to handcraft every detail of the environment model. And from a biological standpoint, this framework offers a quantitative lens to compare seemingly disparate species. It unites chemotaxis in bacteria with olfactory search in flies under one mathematical umbrella.
So what's the long-term impact? Will researchers start measuring these flows routinely?
I think so. The authors provide clear computational recipes and even open-source code for estimating phi from raw trajectories that lowers the barrier for experimentalists to apply this method to their own data, whether it's tracking zebrafish larvae or human gait.
It also invites new questions, like how learning or adaptation might modify these flows over longer timescales.
The paper opens a door to exploring how organisms tune their sensory and motor asymmetries in response to changing environments, perhaps even leading to evolutionary insights. Thanks for joining us on this close look into the paper. We hope you found it as enlightening as we did.
Stay curious, keep exploring the science behind the moves, and tune in next time for more fascinating insights from the world of research.
Until then, keep listening, stay inspired, and remember every movement tells a story. Goodbye.